Hi everyone, my name is Love and I'm one of the SIs for Dr. Maduro's BIO005A class. And for this video, we're going to do part 2 of the practice midterm we went over from the last video, just for a little review of the midterm on Friday. So let's get right into it. Number 15. So what it's asking is, which of the following is not directly involved with the cytoskeleton? Well, let's go over what is involved in the cytoskeleton. Well, we know there's three major types of fibers, right? We got microfilaments. I apologize for my handwriting just from the beginning because it's gonna get bad. We got microfilaments. We got intermediate filaments. And we also got microtubules, right? I'm sorry for the handwriting once again. But we did a more in-depth review of the cytoskeleton in the topic 6 video or 7 video, so make sure you check that out if you need more review. However, let's go into what this problem is asking us. So these three are the fibers that make up the cytoskeleton, right? And they're all proteins, and they're made up of subsets of proteins. Let's go over which proteins each one's made of, right? So we know microtubules, it's in the name. It's made of a protein called tubulin, right? There's different types. There's alpha tubulin, there's beta tubulin. We also know that intermediate filaments are made up of a protein called like keratin, right? And finally, we know microfilaments are made of actin, right? One of the smaller ones. And so which of the following is not directly involved? So let's see what's not in this list that's in the answer choice. So we have microfilaments. We know that makes up the cytoskeleton. We know tubulin is a type of microtubule, so that cannot be it. Keratin is a fiber of intermediate filaments. And we haven't really learned fibronectin yet, so that's probably going to be our answer in this case, right? And then E says all the above, which is not true, all right? Number 16. Number 16 asks us, which of the following graphs correctly shows a relationship between cholesterol concentration in a membrane and its overall fluidity at high temp? So a key thing to remember is high temperature. So as we increase, so basically a membrane, think of it as like butter, right? As you heat up butter, it becomes really liquidy. However, as you cool it down, it becomes a solid, right? If a membrane did that, it would collapse or it would freeze, not allowing things to pass through, right? Which is very problematic. So we want to keep membranes at an optimal fluidity, right? So let's see what happens with cholesterol and fluidity. Well, a little bit of cholesterol or a moderate amount is good, right? It helps us keep fluidity at, or keep us a little solid at high temperature, right? Because if we didn't have cholesterol, then our membrane would be too fluid and it would collapse. So a little bit of cholesterol helps us keep it more solid. However, the more cholesterol we add, the more solid our membrane becomes, right? So as we increase cholesterol concentration in this case, so this is cholesterol and this fluidity, we're going to lose fluidity, right? Because we're adding more and more cholesterol into a membrane, it's going to clog it up essentially, right? So our graph should look something like this, where as we increase cholesterol concentration, fluidity of the membrane decreases. And in this case, we see this is represented by answer choice E, all right? Let's go on to the next one. Or actually, just a quick little review for the last one. If it didn't say high temperature, we'd probably like look for a different type of graph. In this case, since it's high temperature, high fluidity is what we want to kind of avoid, making it too fluid, right? Make it more solid. But too much cholesterol will make it too packed. Anyways, number 17 is asking a person with disease cystic fibrosis. So cystic fibrosis works through a receptor, a gene, which is a CFTR gene, right? And that gene produces a receptor on the plasma membrane called CFTR, cystic fibrosis receptor, right? So transport receptor, cystic. So basically, what it does is it allows chlorine ions to pass through the membrane efficiently. However, if you have a mutation in the CFTR gene, right, because essentially receptors are a type of protein, right? And so they go under the central dogma, basically, right? You go from DNA to RNA to a protein, right? And our protein in this case is the CFTR receptor. So when we make a function, if we have a perfectly fine DNA, that means we'll make good RNA, we'll have a perfectly folded protein, and we'll have a working receptor, right? And in this case, chlorine ions will be able to leave the cell and everything would be healthy. However, when you have a mutation in the DNA or incorrect folding, right? Something goes wrong somewhere along these lines, you'll have a dysfunctional CFTR receptor. So it kind of looks like this, or it's just dysfunctional, right? So now that these chlorine ions were used to pass through, if it was normal, cannot pass through. So when you have a buildup of chlorine ions, this causes a buildup of mucus and essentially causes cystic fibrosis. So let's go over the answer choices and see which one makes the most sense. So A is a person is unable to produce a functioning ion channel. Well, this is true, right? So if you have cystic fibrosis, the disease, your channel, which is this transport receptor, right, is not really making a functional protein channel for these chlorine ions to pass through. So if you cannot go through, then you're going to have the disease. So A sounds pretty correct. Let's keep moving forward. B has a problem with lung membrane phospholipids. Phospholipids has to do with the membrane, not really the protein that we're generating. So it doesn't really make sense here. Cannot make secreted proteins in the rough VR. This could be a possible issue, but we know that the disease cystic fibrosis comes from a defect in the ion channel, right? And this protein is not secreted, it's actually secreted to be embedded into the plasma membrane. D has the disease due to an immunodeficiency. It has nothing to do with immunodeficiency, it's a genetic disorder. E must have at least one parent with the disease. This could be true, but not all the time, because if you just have a defective gene where one of the alleles is like a dominant one, it would cause a defective ion channel. So we'll stick to A in this case. All right, so let's move on to number 18, which occurs with both pyruvate oxidation and glycolysis. Well, let's talk about what happens in glycolysis first. So glycolysis, we are taking glucose, right? So glucose, and we're making it pyruvate, into a pyruvate, right? And basically, I'm so sorry for my spelling and handwriting. It's just hard to type with the mouse. But anyways, we release a lot of things, right? We release ATP. We also release NADH, which is what we're talking about in this case, right? Or possibly talking about, right? We release NADH. And the way that happens is NAD plus is actually reduced to NADH, right? Since we're breaking down a larger molecule glucose into a smaller molecule pyruvate, we're losing some hydrogens, right? And those hydrogens and electrons can be carried through this electron carrier, NADH, right? So that's how that's happened, and that's through a reduction process. This glucose is being oxidized, right? And yeah, so those are two major things. So ATP, NADH, and pyruvate. Let's talk about what happens in pyruvate oxidation. Well, we're going in with pyruvate, and we're coming out with acetyl coenzyme A, right? Key thing I'm going to keep repeating is that every time we lose a carbon, we're releasing it as CO2 in glycolysis, right? So we're going from a 3-carbon to a 2-carbon, right? So what comes out of it? Well, we get a CO2, and we're also getting an NADH being NAD plus being reduced as well, right? So we're going to have another reduction process, right? So now let's look at the answer choice as well. We know what occurs. So we know what occurs in glycolysis, right? We get an ATP, but we don't get that in pyruvate, right? So A, unfortunately, will not work. B, NAD plus is reduced. That we know is a fact, right? So we know that NAD plus is reduced in glycolysis, and it's also reduced in pyruvate oxidation. So B is a possible answer choice. C, CO2 is made. Well, in glycolysis, we're going from a 6-carbon chain to 2 3-carbon chains, right? So in the end, we're still preserving 6 carbon, so no carbon dioxide release. Pyruvate oxidation, we are losing a carbon because we're going from 3 to 2, right? So unfortunately, only one of them does that. D, oxygen is needed. Remember, glycolysis occurs in the cytosol where no oxygen is needed as well. And if oxygen is not present, then it goes to fermentation. If it is, then it will continue cellular respiration in the mitochondria. So unfortunately, that's not the case. And ATP is hydrolyzed. Only glycolysis uses ATP. If you remember, there's 2 stages to it. There's the energy investment, and that's where it is. However, pyruvate does not require any of that. So unfortunately, our answer is only B. Okay? Number 19. What is delta G for the reaction whose profile is shown to the right? Well, key thing to remember, when we talk about delta G, we're just looking for the difference between the reactants and the products, right? In our case, we started at -60, as we can, we started at 60, not negative, and we're dropping down to 10, right? So the formula is delta G, products minus reactants. So it equals products minus reactants. So in this case, 10 minus 60, and we get, in this case, -50, okay? Which gives us B. So it's always gonna be the difference between our products and our reactants. This graph could look the other way around. So this is an exergonic reaction, right? It's negative. However, sometimes you may see something that looks like this, right? And in this case, you'll have a positive delta G, right? Because the difference from here to here is a positive number, right? That's endergonic. So let's move on to 21. 21 is saying that a drug X can get into the cell membrane and allow diffusion of charged particles from one side of the membrane to the other, right? Key word here would be diffusion. If this drug is given to muscle cells during cellular respiration, which of the following would most likely happen in the mitochondria as a result, all right? So what we're saying here is that a drug basically allows the diffusion of charged particles from one side of the membrane to the other. And what we know about charged particles when we're talking about cellular respiration, let's think of protons, right? The electron transport chain. All right, so let's talk about what happens in a normal electron transport chain, right? We have our outer membrane here. We have our inner membrane here, right? And in the middle, we have something called the intermembrane space, right? So I'm gonna call it inter. And essentially NADH over here, NADH gets oxidized, right? And we release proton, and we basically kind of just pump them on using electron energy to this side, right? So we have a bunch of H's here. And what we're doing is we're putting them against the gradient, right? Because we're trying to create a high concentration of protons here in order to use a proton motive force to come down here to make ATP in chemiosmosis. However, if we look at what this drug is doing, it's allowing us to do diffusion of charged particles, right? So essentially, it's gonna take out the functionality of our electron transport chain. So what's gonna happen is if we build up a high concentration in the intermembrane space, and there's a low one here, and we throw this drug in, well, all these protons will wanna go through equilibrium, right? So what they're gonna do is wanna come back in, right? So let's change the color to like blue. And so they're gonna actually wanna come back in when we put this drug, right? From the intermembrane space to the matrix, which is over here, right? So let's look at what answer choice encompasses this description. So 21, protons would flow directly from the matrix to the intermembrane space. Well, remember during normal cellular respiration, this is actually active transport, right? It's not diffusion. And we're trying to create a high energy concentration area here of protons. So this drug would actually do the opposite. So that's not our answer. B, electrons would flow directly from the matrix to the intermembrane space. So basically electrons, as we know, flow through the inner membrane, right? Through a series of proteins, right? So that wouldn't have much to do with the matrix or the intermembrane, right? In this case. Protons would flow directly from the intermembrane space to the matrix. Yeah, so this one sounds pretty good to us, right? We're going from a high concentration of protons to a low concentration over here. Finally, D, electrons would flow directly from the intermembrane space of the matrix. Once again, electrons are usually stuck in the inner membrane, right? And they basically go, so say this is our inner membrane, and basically they go in, travel through a series of proteins, and come out back in the matrix, where O2 is the last acceptor, and it makes water, right? So in this case, D was not a good answer for us. And E, nothing different would happen. No, we obviously know that if this drug causes diffusion, we know that there's some issues going on here with not equilibrium and concentration of protons. So this drug would force that equilibrium to happen, and protons would move to the matrix, right? So number 22 is asking us about the endomembrane system and which of the following organelles are part of, or membranes are part of the endomembrane system except. So what I like to think of when I think the endomembrane system is basically a secretory protein route, right? So what is involved? Well, we have a nucleus, right? We also have the ER, right? We also have the Golgi, right? And we also have lysosomes, right? Because those Golgis can be transported in vesicles through lysosomes, right? Through and vesicles, right? So I usually like to think of that route when I think of the endomembrane system. Mitochondria, right away, that should be a red flag, right? That it does its own thing, right? Mitochondria, think of it as like its own organelle. It was an endosymbiont, right? So that way it's not really interconnected with our general nucleus and those type of membrane brown organelles. And essentially it does its own thing, right? It has its own DNA, it has its own ribosomes. So it's not really part of the endomembrane system in this case. So that's probably our answer. Lysosomes, as we said, secretory route, we do send stuff to lysosomes. Golgi is connected with the ER pathway. Smooth ER and nucleus are also connected together. So in this case, our answer is A. So number 23 is asking about insulin. So first thing, let's just do a mini review on insulin. What do we know about insulin? Number one thing, insulin is a protein, right? So how is that made? Well, DNA is transcribed to RNA. RNA is translated to a protein. And that translation pathway is a secretory pathway, right? So the mRNA will come to a free ribosome. That free ribosome will translate the signal sequence. That signal sequence will say, hey, I need to go outside of the cell. So it would bind to the ER, and it would be a bound ribosome. That's where it'll finish translating. And then it'll be packaged and shipped out from the Golgi outside of the plasma membrane into the bloodstream, right? How does it work? Well, insulin is essentially a ligand for an insulin receptor, right? In most cases, like a GLUT4, we call it, right? And when it binds to the receptor, channels open up, allowing glucose to come through into the cell in order to be broken down, right? If something is malfunctional in this area, then glucose stays in the bloodstream, and that's where we see people that are diabetic, right? Because their glucose cannot be broken down. Essentially, let's answer this question. Insulin is a channel protein for facilitated diffusion of glucose. Insulin is not the channel protein. It's basically a ligand. It's a hormone, right? That's a number one thing as well. It's a hormone, right? So it binds to a receptor. Nope. B is a secreted enzyme that digests glucose in our blood. We know it's not an enzyme, right? It's a hormone. And number two, we know that glucose is digested through a series of other enzymes in glycolysis, all right? In our cells, not our blood either, all right? C, binds to glucose and brings it into cells by endocytosis. It doesn't bind to glucose, it binds to a receptor, right? That allows glucose to come in through a different channel, okay? So this is not the case. D, causes cells to take in glucose using passive transport. Exactly. So this is essentially what we're talking about, right? So let's kind of break it down. So say this is our bloodstream, and we have a bunch of glucose. So these dots are the glucose, right? Say this is my cell right here. I don't have glucose inside. So general rule of basically what we learned, right? Diffusion, right? High concentration, low concentration. If I have a high concentration of glucose, it will passively come into the cell if I open up a channel to, right? So what happens is insulin, which I'll draw in blue, essentially binds to an insulin receptor saying, hey, open up the channel. Once the channel opens up, simple diffusion occurs, right? High concentration of glucose in the blood. There is none in the cell, low concentration. It will come in on its own, right? Passive transport. So D is our perfect answer. E, insulin is a steroid hormone that binds to an insulin receptor. It's not a steroid hormone. It's a protein hormone, essentially. All right. So yeast cells are growing in the presence of oxygen and glucose. All right. So essentially, it's telling us cell respiration, right? That should be going off in our heads when we hear that. And it's saying the chemical cyanide permanently blocks electron flow in the mitochondrial electron transport chain. So basically, electron transport chain will be inhibited. What would the rate of oxygen consumption look like over time t with the treatment of cyanide? Well, we know cyanide only hits us when we enter the electron transport chain, right? So we know that we go glycolysis, right? Right? And then this is the O2 stage. If we have O2, we will go PO, pyruvate oxidation, we'll go CAC, citric acid cycle, and then we'll go ETC, right? So essentially, we're gonna talk about O2 consumption starting from here till the end over here, right? And what we know is that this drug is gonna block O2 consumption over here. But all this is still okay. So if we were to draw a graph, all this is still stable consumption of oxygen, right? However, if we don't have the ETC, we don't have that last carrier, electron carrier, we don't have oxygen as its last electron acceptor, and there would be no use of it, right? So essentially, oxygen will not be used, and it will go to 0. So a graph that we're talking about looks more of like something that we see in C, right? A is still showing somewhat of a consumption till the very end, which doesn't really make sense, right? If you can't use it in the AETC, then you can't really move on. So A is not right. B shows like a stable route and even higher consumption, which doesn't really make sense. D saying even higher, which doesn't make sense. E is showing completely stable, but we know that it gets interrupted at the end. So that's not a fact, right? The answer in this case is C. Which of these are in an oxidized state? Okay, so this one, I would say the best way to think about it is refer to cell respiration. So let's go to CO2. Where do we know CO2 is made? Let's think of pyruvate oxidation, right? So we know that we're going from a 3-carbon chain and it's going to a 2-carbon chain. Now, a fact that you know, a thing that you've heard consistently throughout this topic is that NAD plus, my bad, gets reduced to NADH, right? So, reduced. Now, if something gets reduced in a reaction, that means something else had to get oxidized. In this case, we know pyruvate oxidized a carbon dioxide, or oxidize a carbon into carbon dioxide, right? We're taking out a CO2, right? So that is definitely one of our answers, right? Because in pyruvate, our carbons do have hydrogens, and we are removing them to make this carbon dioxide. So you can think of it that way. But best way to think about it, if something gets reduced, something else gets oxidized. So we know that CO2 gets oxidized in this case. Now let's talk about so we can knock out answer choice anything, or we can keep anything that has answer choice 2 in it. So C is out and D is out. So we have a choice of A, B, and C, or A, B, and E. So let's talk about water and oxygen, all right, because those are pretty similar in relativity. So when we have the very end, right, we're talking about O2. Now, O2 exists on its own, right? While water, you added hydrogens to O2, right? So essentially, O2 gets reduced to become water, right? The very last electron acceptor in the electron transport chain is oxygen, and it reduces into water, right? So if water is the reduced state, then what got oxidized? Well, before that, it was oxygen, right? So that would be our answer in this case. So 2 and 3. And that leaves us with answer choice E, right? So this is it for our video for today. Good luck on your test on Friday. Study hard and hope for the best. Thank you.